So our group, well, my group, is the fertility, pregnancy, and childbirth, and more specifically the vaginal microbiome. My name is Steven, and I'll be giving a quick summary, sort of, on the paper titled "The Ovarian Cycling and the Reproductive State Shape the Vaginal Microbiota in Wild Baboons." So the first, I guess, author is Elizabeth Miller. She is currently a research associate and postdoctoral researcher at the University of Minnesota. She obtained her PhD at Notre Dame and her Bachelor's of Arts at Oberlin College. Uh, some of her expertise include pathogenomics, microbial ecology, antimicrobial resistance, and microbiome. I'm not sure if I'm pronouncing those correctly. Sorry if I don't. Second professor or author that I found information on is Elizabeth Archie. She is currently the professor of behavior and ecology and disease ecology at the University of Notre Dame. She obtained her PhD at Duke and her BA at Bowdoin College, both for biology. And some of her expertise include behavioral ecology, microbial ecology, disease ecology, and evolution and eco immunology. Yeah, I'm not sure if I'm pronouncing that correct either. The main question that shapes this paper is whether or not the shape of the human vaginal microbiome is unique to humans themselves, or is it shared with other primates or mammals? Since we know a little about causes and consequences of inter-individual variation in the vaginal microbiome of non-human mammals, this question helps us understand the general factors that come into play for inter-individual variations in the vaginal microbiome in different species and how it can be useful models for vaginal microbial communities overall. There's a huge contrast between humans and non-human primates vaginal microbiota. For example, non-human primates have few lactobacilli and higher taxonomic diversity than humans. Um, the main objective in this experiment was to test the effects of reproductive state and ovarian cycle phase on the microbial composition of the baboon vaginal microbiome in order to understand whether the reproduction related changes in baboon vaginal microbiota are parallel to humans. Um, the study experiment was on 48 wild female baboons. During the study, they also tried to explore if other factors such as age, kinship, dominance, and rank, and social group size may affect the inter-individual differences in baboon vaginal microbiota. Biota. They collected 52 vaginal swabs from the baboons across different reproductive states and stages of ovarian cycling, such as like cycling, pregnancy, and lactation. They then use 16S rRNA genetic sequencing in order to identify and analyze the composition of the bacterial communities that are in the vaginal swab samples. They compare the composition of the vaginal microbiota across the different reproductive states and stages of ovarian cycling. Um, During this experiment, they also researched and tested other factors, including the alpha and beta diversity, the horizontal and vertical transmission, and the determination of vaginal pH levels. They found that the alpha and beta diversity are different in the vaginal microbiota depending on the stages of the reproductive states. For beta diversity, the vaginal microbiota was significantly different between baboons in estrus than baboons in other phases, while for the alpha diversity, it is higher in the luteal phase than in any other phases. The two modes of transmission are horizontal and vertical. Horizontal is between individuals, so just like one baboon to like another baboon, while vertical is from the mother to the offspring. They found that infant baboons have a higher abundance of lactobacillus species, similar to their mother's microbiota, which showcases how important vertical transmission is to vaginal microbiome in baboons. Vaginal pH is important in determining how to maintain a healthy vaginal microbiota since certain bacteria thrives in different pH environments. researchers found that baboons and estrus had a lower vaginal pH compared to baboons in other reproductive states. They also discovered that baboons with a lot of lactobacillus species are correlated with lower vaginal pH levels. Um, as a whole, study suggests the vaginal microbiota in the baboon differs depending on the reproductive state and stages of ovarian cycling. Despite the contrast difference between humans and non-human primates, the reproductive state was the strongest predictor of the baboon vaginal microbiota. Additionally, they saw similarities between humans when baboons were in the ovulation phase. This included the highest levels of lactic acid producing bacteria, which predicts the lowest vaginal pH. The findings suggest that the vaginal microbiota of humans and baboons are under similar selective forces, which indicates that these forces may be fundamentally important to many other mammals. Overall, the study suggests that changes in the vaginal environment, including pH and hormone levels, may be important in shaping the composition and diversity of the vaginal microbiota in baboons. These findings have important implications for understanding the role of the vaginal microbiota in reproductive health and fertility in other animals, including humans.
Hi, my name is Zoe, and I will be talking about the next paper, which is called The Maternal Vaginal Microbiome Partially Mediates the Effects of Prenatal Stress on Offspring Gut and Hypothalamus. So the primary investigator of this research, her name is Dr. Tracy L. Bale. She originally got her bachelor's in biology and molecular biology and genetics from Washington State University. She went on to get her PhD in neurobiology from the University of Washington. Then she was a postdoctorate fellow in neuroendocrinology at the Salk Institute. And now she is a professor of pharmacology at the University of Maryland College of Medicine. She's most known for her research on stress as a risk factor to neurodevelopmental disorders and neuropsychiatric disease. Dr. Bale's lab has developed mouse models to study vulnerability to stress dysregulation, assessing sex specificity, developmental timing, and epigenetic mechanisms involved in the programming of the brain, placenta, and sperm in response to stress. The next researcher I'm going to address is Eldin Yasarevic. He is a PhD in neuroscience from the University of Missouri, Columbia. He is now an assistant professor in OBGYN at the University of Pittsburgh, but he was a postdoctorate fellow in the Bale Lab at the University of Pennsylvania, where this research took place. He also has many similar publications like prenatal and postnatal contributions of the maternal microbiome on offspring programming, and childhood adversity impact on gut microbiota and inflammatory response to stress during pregnancy. So at the time that this study was released, reports had recently shown that there is an association between the developing brain and bacterial microbiome within the gastrointestinal tract. The mechanism of this was not yet understood, but previous studies have shown that the initial maternal microbiota that colonizes the gut of an offspring has a lasting effect on the brain and the GI tract going forward. Previous studies have also shown that maternal stress during pregnancy alters the vaginal microbiome, which can then be transferred to offsprings at birth. Using a mouse model of early prenatal stress, the same researchers as this paper in a previous study were able to confirm that the maternal prenatal stress alters both the composition of the vaginal microbiome as well as the microbiome of the neonatal gut. They saw altered metabolite profiles in the gut and hypothalamus of the offspring. They also saw reduced body weight, an increased neuroendocrine response to acute stressors, increased anhedonia, which is reduction in the ability to experience pleasure, as well as altered stress-related gene expression. All of these aspects were seen in male but not female prenatal stress-exposed mice. No effects of prenatal stress were seen in the female offsprings. This new study takes this information one step further in order to ask the question, does the maternal vaginal microbiome directly mediate the effects of prenatal stress on the offspring? So in order to answer this question, the researchers needed to control for a few different effects, the effects of the postnatal environment and of the housing conditions. To do this, they performed cesarean sections on both control maternal mice and on stress-exposed maternal mice in order to prevent exposing the offsprings to the vaginal microbiome at birth. The colonization of the microbiome in the offsprings was then reproduced by exposing the neonate to the maternal vaginal fluids at birth after being collected from the mothers by implanting an orogastric gavage to deliver the maternal vaginal fluid to the mouse pups. There were 4 experimental groups: the control mouse offsprings that were inoculated with the control mouse microbiome, or CC, control mice; control mouse offsprings inoculated with the stress-exposed microbiome, which is SC mice, so control mice inoculated with the stress-exposed microbiome. There were stress-exposed offsprings that are inoculated with the control microbiome, or CS mice. And finally, there were stress-exposed offsprings inoculated with the stress-exposed microbiome, SS mice. All mouse pups were then fostered with surrogate dams in order to control for the transmission of microbiome through living exposure. Then, using a gene sequencing approach to compare the gut microbiota, the researchers observed differences in the microbial community composition and abundance between the different experimental groups. Notably, they observed differences in the abundance of E. coli, S. acidominimus, and S. thoraltensis between the CC and SC offsprings. As a refresher, the CC group are the control offsprings that are inoculated with the control microbiome, while the SC group are control offsprings that are inoculated with the stress-exposed microbiome, which reveals that colonizing control offspring with a vaginal inoculant from the stress-exposed dam resulted in a gut community composition that was not different from the SS offspring. In other words, inoculation of these control mice with the stress biome caused their gut microbe to resemble the stress-exposed offsprings that were inoculated with the stress-exposed microbiome, proving this hypothesis correct. The next concept to be explored was how the difference in the microbiomes affect body weight and stress responses in the offspring. Measurements from postnatal day 14 to postnatal day 70 showed that the body weight growth curve for SS male offsprings were lower than that of CC male offspring. The body weight of the SC males was not significantly different from either the CC or the SS males, showing that there was an intermediate body weight phenotype in the SC males inoculated with the microbiota from the stress-exposed dams. This is seen in figure 2A. As in the researchers' previous model, there were no differences observed in the body weight of the female offsprings. And in terms of this class and the background that we have learned, the most important findings, I would say, were when the researchers explored differences in the HPA axis response. To do so, acute stress-induced corticosterone levels were measured after a 15-minute restraint session to stimulate a stress response. They observed that these levels were significantly higher in prenatally stressed males compared with control male offsprings. This can be seen in figure 2C. However, again, there was no effect on corticosterone levels in female offsprings. When comparing all of the groups, the CC and the SS groups showed the same as in figure 2C. Corticosterone levels in response to restraint in males SC offspring were increased compared with the CC offsprings. But they were not significantly different from those of SS males, suggesting that an intermediate phenotype, as seen in the body weight measurements, and a possible mediating effect of the maternal vaginal microbiome on this aspect of prenatal stress phenotype. Also importantly, male CS offspring had a stress response that was higher than CC offspring, and that did not significantly differ from the SS offspring, which suggests that the inoculation of a control maternal vaginal microbiome was not able to rescue this aspect of the prenatal stress phenotype. Once again, no differences were detected in the female offsprings of any experimental group. So for some of the main points, epidemiological studies have reported that there are associations between C-section and increased risk for obesity, asthma, and allergies, suggesting that exposure to vaginal microbiota during birth may be linked to long-term health outcomes. This study seeks to explore the causal relationship between maternal stress exposure and impact on offsprings by way of the vaginal microbiome. The stress-exposed offsprings were mirrored in the control group inoculated with the stress-exposed microbiomes in terms of body weight and cortisol levels. However, the stress-exposed offsprings inoculated with the control microbiome did not show a rescue of stress effects on body weight and cortisol response. This leads to the conclusion that maternal stress has effects on the offspring that are partially mediated by the maternal vaginal microbiota, but that cannot be rescued by it. The gut microbiome has several purposes, including mediating the immune system. The researchers suggest that more research is needed into specifically examine the impact of an altered gut microbiota on fetal immunity. Overall, the data suggests that prenatal stress influences the fetal gut as well as the hypothalamic gene expression, which could possibly play a role in the increased risk of neurodevelopmental and gastrointestinal disorders.
Hey guys, I'm Maria, and I am going to be presenting the final paper for our narrative, continuing the topic of the vaginal microbiome and its impacts on pregnancy, amongst several other aspects of the female body. So the paper I'm discussing regards the rectal and vaginal eradication of Streptococcus agalactiaceae, also known as GBS, in pregnant women by using Lactobacillus salivarius CECT 9145, a target-specific probiotic strain. So I'll start by introducing the authors, first of whom is Virginia Martin, a PhD. She is on the Spanish National Research Council, and she is part of the Department of Nutrition and Food Science at the Complutense University of Madrid. She received her education from Institute of Physical Chemistry, Rocca Solano, where she received her PhD. And then I also wanted to include a prominent author of the study, Juan M. Rodriguez. Unfortunately, I could not find a picture of him, but he is a faculty member at the Department of Nutrition and Food Science, also at the Complutense University of Madrid. Also, just wanted to include Nivea Cardenas with her PhD because she had a major role in the paper as she was responsible for the collection of strains and laboratory analysis, which was a big part of the paper since it was analyzing microorganisms of the vaginal microbiome. She is part of the Department of Galenic Pharmacy and Food Technology at Complutense University of Madrid, and she received her education from there, and she also has her PhD in veterinary science. So I first wanted to start out by introducing the Streptococcus agalactiaceae strain, also known as the Group B Streptococci, since it is one of the microorganisms most frequently involved in severe neonatal sepsis cases, which is very detrimental, and the reason this paper is being written and this is being studied is to be able to eradicate this, since also it has been found in systematic reviews and meta-analysis that the estimate for maternal GBS colonization worldwide is 18%. And it is considered a major risk factor for ascending infection during pregnancy, which is not very ideal. Current preventative measures include intrapartum antibiotic prophylaxis, but it does have several limitations, which include that it does not guarantee GBS eradication, it does not prevent GBS-related abortions, stillbirths, and preterm births. It may lead to increasing rates of antibiotic resistance among GBS and other clinically relevant microorganisms, and it has a very negative impact on and a lasting effect on the host's health and the development of the microbiota of the infant as well. So very harmful as a preventative measure. So now I'm going to introduce Lactobacillus salivarius CECT 9145, which is a probiotic strain that will be used for the eradication of GBS. It was found to have in the study, it was found to have antimicrobial activity against GBS, which is what we wanted. And it showed production of specific antimicrobials such as bacteriocins, lactic acid, hydrogen peroxide, which all were aspects that inhibited GBS growth. Also, it was shown that the genus Lactobacillus constitutes the dominant bacterial group of the vaginal tract already in most healthy women. So it's a natural microorganism playing a key role in genitourinary homeostasis. So as they continued the study, the microbiological analysis of vaginal swabs of the participants, which were obtained from pregnant and non-pregnant women, isolated a particular strain of lactobacilli, which was what was shown to have this antimicrobial activity. Also, previous studies have shown the ability of lactobacilli strains to inhibit the growth of vaginal pathogens. So this is why it was a key strain to be looked at for this particular study. So in addition to its antimicrobial activity, it did show co-aggregation and co-culture assays with the GBS S. agalactulae, which is what we wanted to see to show that it will attack those microorganisms. And finally, it showed adhesion to vaginal cells and to mucin, which was what was considered, which was preferred as well, so that it shows that it is effective. Along with that, the lactobacilli showed survival in vitro and oral toxicity in the rat model was also tested for. So survival after in vitro exposure to saliva and gastrointestinal-like conditions was seen, which is ideal. And with the survival, we are able to see that it is able to live in the gastrointestinal environment of the vaginal tract. Also, a rat model was used to test oral toxicity to make sure it was safe, and all animals survived both oral toxicity trials in the paper. And the development of the treated animals during the experimental periods corresponded to their species and age. So there was also no significant differences in body weight or body weight gain among the groups treated with L. salivarius, the lactobacilli strain. As you can see in this table, to go back to the point of survival, it shows all these percentages are the different strains of lactobacilli that were able to survive in conditions simulating the gastrointestinal tract, which were pretty high, so that's good. Finally, the major takeaway and key finding of the paper was the efficacy of GBS eradication from the intestinal and vaginal tracts of pregnant women. And this was a pilot trial, so this means that it was one of the first to be able to be mirrored in human women, pregnant women. The study had 57 participants who were pregnant women, 39 were rectal and vaginal GBS positive women, 18 rectal and vaginal GBS negative women at the start of the intervention. They were aged 25 to 36, and all of them met the criteria of healthy status. And volunteers were distributed into 3 groups, 1 probiotic group and 2 placebo groups. Probiotic group was was GBS positive, and they were given the probiotic, which was the lactobacilli, and the 2 placebo groups, one of which included GBS positive women that received IAP, which was the current preventative measure that had its limitations, as we discussed in the beginning of the paper, and they had been receiving this because their baby had suffered a GBS sepsis already, and these were women that were given IAP if their babies suffered from GBS sepsis. And the second placebo subgroup included GBS negative women as well, without IAP. So it was another control group. So women in both of these placebo groups received the excipient that carried the probiotic strain in the form of milk, and recto-vaginal GBS screening was performed at different points across several weeks. Results were promising, as they showed significantly that the group of GBS positive women that started using the probiotics since they were enrolled in the study also tested positive for GBS at 28 weeks, but an increasing number of GBS negative results appeared in the successive swabs collected until delivery. As you can see in this table, it is showing a successful decline in GBS positivity, showing that it was being eradicated by the lactobacilli probiotics, which was promising. And it was finally concluded that the administration of the L. salivarius lactobacilli CECT145 strain specifically, which was administered to GBS positive pregnant women, is safe since it did not show any adverse effects, and it is a successful strategy to significantly decrease the rates of GBS colonization during pregnancy, which in turn, consequently, may cause neonatal sepsis. So this definitely plays a role in a lot more neonatal survival. So that is it for our presentation. I'm looking forward to discuss tomorrow. Thank you.